All right. Next 4 questions. What are the components of pyruvate oxidation? So remember, step 2 is pyruvate oxidation and the Krebs cycle. Question after that, how are the electrons transferred during oxidative phosphorylation? How is energy produced during oxidative phosphorylation? And what about when oxygen is absent? And you all know from doing lab this week, all of you were in lab this week, I think. No, not all of you were in lab this week. Most of you were in lab this week, and we talked about fermentation, right? When we force yeast to metabolize sugar in the absence of oxygen. When oxygen can't diffuse in there rapidly enough for them to be able to do aerobic respiration, what happens? Okay. All right. So first question, what are the components of pyruvate oxidation? Pyruvate oxidation. All right. So we talked about this already, but pyruvate oxidation and the Krebs cycle take place where? Inside the mitochondrial matrix. So pyruvate has to be transported into the matrix where there's a lot of it waiting for access to an enzyme to facilitate its oxidation. And so we're transporting pyruvate from where there's little of it into a place where there's a lot of it. We're transporting it against its concentration gradient. So it has to be actively transported. And we know that that requires energy. We also know that that requires a protein specific to that process. And so once it can get into the matrix, it can be oxidized into carbon dioxide and the acetyl group for acetyl-CoA, which needs to get put into the Krebs cycle. Okay, so ultimately we take pyruvate, we oxidize one of its 3 carbons into carbon dioxide, and the other 2 remain as an acetyl group to be attached to coenzyme A. Now, this process of pyruvate oxidation does not produce any ATP. Okay, oxidizing pyruvate into the acetyl group of acetyl-CoA does not produce ATP. It does, however, produce 1 additional molecule of NADH. So glycolysis produced 2 molecules of NADH, right? Right? Yes. And now pyruvate oxidation produces another 2, because keep in mind, we make 2 molecules of pyruvate with every 1 glucose molecule, right? So if each pyruvate molecule generates 1 NADH, we generate 2 of them for every 1 glucose, because 1 glucose gives us 2 pyruvate, each pyruvate gives us 1, and 2 times 1 is 2. Beautiful. And so here's the overall process of pyruvate oxidation. Now, for a lot of these reactions we've had so far, I've told you, this reaction doesn't actually happen, but rather is an overview of several reactions. This one's a little different. This reaction actually happens, okay, where we take pyruvate and coenzyme A and nicotinamide dinucleotide, and we produce acetyl-CoA, NADH, a hydrogen ion, and 1 molecule of carbon dioxide. This reaction actually takes place to oxidize one of the carbons from pyruvate. Right? So it's a little different. The overall reaction of cellular respiration doesn't happen. If it did, life would be a little bit more interesting, right? If you're actually burning sugar in the presence of oxygen in your cells, that'd be pretty cool. I think cells would need to be built differently to be able to handle that amount of energy being produced that quickly, but it'd be cool. But that overall reaction doesn't actually happen, right? It's a summary of everything that happens during cellular respiration. We saw this also for glycolysis, right? The overall process of glycolysis. It's 10 reactions, but we can represent it as like a single process. But this actually happens. Now, acetyl-CoA, which are 2 of the 3 carbons that were in pyruvate remaining attached now to coenzyme A, it enters into step 2B, right? Pyruvate oxidation is step 2A, Krebs cycle is step 2B, where we oxidize the rest of the carbons from pyruvate. 1 of the 3 carbons oxidized in this reaction, the other 2 are going to be oxidized during the Krebs cycle. All right. So the remaining carbons in acetyl-CoA are oxidized into carbon dioxide during the Krebs cycle or citric acid cycle or TCA cycle, whatever you want to call it, 3 names for the same thing. If we are going to oxidize carbon dioxide, what are we releasing? Oxidation is loss of what? Electrons. Something needs to be reduced as carbon is being oxidized, right? Something needs to be reduced as carbon is being oxidized, and we reduce 3 molecules of NAD+, nicotinamide dinucleotide, and 1 molecule of FAD, every time we do the Krebs cycle. Every time we do the Krebs cycle. How many times do we do the Krebs cycle for every 1 glucose molecule? Twice. Which means for every 1 glucose molecule that goes through the Krebs cycle, we get 6 more NADH and 2 FADH2. Okay? Oh yeah, here it is. Each turn generates this. We get 2 turns per glucose molecule, because every 1 glucose molecule gives us 2 pyruvate molecules, and every 1 pyruvate gives us 2 acetyl-CoA, right? Every 1 pyruvate gives 1 acetyl-CoA, so 2 pyruvates give 2 acetyl-CoA. I misspoke there. Sorry. It's Friday. It's Friday before a very nice weekend. A weekend that is 7 Saturdays and 2 Sundays. Sorry. It's wonderful. So good. I've never in my life wanted spring break as badly as I want it right now. Never in my life. Man, it is going to be good. You're welcome for the days off you're going to get from this class. All right. So here's another reaction that is not representing an actual reaction, but an overall process. So this is the overall process of doing the Krebs cycle one time. Doing the Krebs cycle one time, we take acetyl-CoA, 3 molecules of NAD+, 1 molecule of FAD, 1 molecule of ADP, 1 inorganic phosphate, and 2 water molecules, and we produce 2 molecules of carbon dioxide, 3 more NADH, 1 FADH2, 1 ATP, 3 hydrogen ions, and 1 coenzyme A that can be recycled and attached to another acetyl group from a future pyruvate. So again, this is a reaction that does not actually happen, but is a summary of the Krebs cycle, which is 8 reactions. Oh yeah, coenzyme A can help other biomolecules enter into the citric acid cycle. So as you metabolize protein, you'll break amino acids down, and amino acids will get attached to coenzyme A and will come into the Krebs cycle without having to go through glycolysis. As you metabolize fats, you'll take fatty acids and you'll hydrolyze them 2 carbons at a time, attach them to coenzyme A, and enter them into the Krebs cycle without doing glycolysis. Yeah.
So, I don't know if you've explained this already or not, but you said that reaction doesn't actually take place.
Correct.
Is it because
this is the overall big picture of what happens during 8 separate reactions.
Okay.
Yeah. Okay. So in glycolysis, I told you 10 reactions. You need to know the 10 enzymes, what you start with, what you end with, and anything extra that happens. For the Krebs cycle, we have 8 reactions, and you don't need to know any of that. Okay? Now, well, I shouldn't say you don't need to know any of that. You need to know the extra stuff that happens, right? You need to know that we're producing 1 ATP every time we do the Krebs cycle, right? Which we're going to do it twice for every 1 glucose molecule. You need to know that we're producing 3 NADH for every 1 cycle, but we're going to do it twice for every 1 glucose. Okay, so you need to know these extras, but you don't need to know the enzymes or what you're starting with or what you're ending with. We'll go Davies and then Levi.
Is this all happening in the matrix?
This is all happening inside of the mitochondrial matrix, yes. Yep. Levi.
So the products basically of this reaction is what we need to know, the basics, like the...
Yeah, yep. like you need to know this overall reaction,
Okay.
right? This overall process of what happens during the Krebs cycle, you need to know this. But you don't need to know the 8 individual reactions. Like, I don't care if you know which reaction produces the 3 NADH. There are 3 separate reactions that each produce 1 NADH. I don't care. This is something you should be able to replicate. This figure here provides, again, that summary of step 2, where we oxidize one of the carbons from pyruvate into carbon dioxide, and then attach that to coenzyme A to shuttle it into the Krebs cycle. And then in the Krebs cycle, we're going to oxidize the other 2 carbons from pyruvate, and we're going to generate 3 molecules of NADH, a molecule of ATP, and a molecule of FADH2. Okay, that is absolutely something that you should know, and that overall reaction. This is something you should be able to replicate. And then this is just kind of focusing in on this portion here. Now, if you are dissatisfied with that, you're sitting there and you're in a position where you're like, Dr. Engel, I want to know this just as well as I know glycolysis. By all means, do the same thing you do for glycolysis. Take an index card, and on one side of it, write the enzyme that catalyzes the reaction. On the other side, write what it starts with, what it ends with, and any accessory stuff, and then practice getting them in order. Okay? You can do that. The reason why I have you know this for glycolysis and not for Krebs cycle is because every organism on Earth, every living organism on Earth goes through glycolysis. Every one. And whether you do aerobic or anaerobic respiration, you go through glycolysis. But the Krebs cycle is only present in organisms that can do aerobic respiration, and is only present when you're actually going through aerobic respiration. That's not to say it's not important, it's just different. And because it's different, there are different requirements and what you need to know. But here's a great diagram to go through and to get all the individual pieces. So here's 1 reaction, 2 reaction, 3 reaction, 4, 5 reactions, 6 reactions, 7 reactions, more, 8. Okay, so there are the 8 reactions of the Krebs cycle. Now, if you look through here, you'll see an enzyme name that to you should spark the memory of it's catalyzing an oxidation reduction reaction, right? And those are dehydrogenase enzymes. So those are catalyzing the oxidation reduction. Every time you see a dehydrogenase, you should see either NADH or FADH2 being produced. But, again, you don't need to know all of these reactions individually. You do need to know the big picture of what happens during the Krebs cycle. Okay, so now, how many ATP do we make during glycolysis? Net ATP. 2. How many ATP do we make during pyruvate oxidation?
2.
Just before we do the Krebs cycle? 0. How many do we make during the Krebs cycle? 1 for each pyruvate, or for each acetyl-CoA, 2 total. Okay, so we've accounted for 4 of the 32 ATP we get per glucose molecule in aerobic respiration. The rest is going to come from step 3. But how many NADH do we make during glycolysis? 2. Yes? We make it in step 6 of glycolysis. We make NADH, and we do step 6 twice for every 1 glucose molecule. So we make 2 NADH during glycolysis. How many NADH do we make when we oxidize pyruvate into acetyl-CoA? 2 more. 1 for each pyruvate, but we have 2 pyruvates for every glucose molecule, so that's 4. How many NADH do we make during the Krebs cycle? 6, right? 3 each time we do it, and we do it twice for every glucose molecule, so that's 6 more. That's 10 total NADH molecules that we make during steps 1 and 2. Yes? 10 total NADH. And when you see NADH, what do you see? 2 electrons. So that's 20 electrons. How many FADH2 molecules do we make during glycolysis? None. No FADH2. How many do we make as we oxidize pyruvate into acetyl-CoA? None. How many do we make during the Krebs cycle? 2. So we have 10 molecules of NADH and 2 molecules of FADH2 to transfer electrons in the step 3. Okay? These are kinds of things that you really need to be able to do. Okay? Just say hypothetically on the next exam, if you were asked a question like this. If so little ATP is produced during glycolysis and the Krebs cycle, why are they so important? There's several ways you can answer that. One, you could say, well, sometimes you can only do glycolysis, right? And so it's important because sometimes you can't do the other steps. Or you could say something like, while it's true that they don't produce much ATP, what they do produce is electrons that can be used in step 3. Okay? Make sense? Maybe? Okay. So here's a nice diagram. And what I want you to do with this diagram is basically this. I want you to be able to look at this diagram and to be able to explain how proteins, carbohydrates, and lipids can all be used as fuel molecules. This is something we talked about on Friday, the first week we ever met together. We went through and we talked about our individual biomolecules, and we talked about that proteins can be both structural and provide energy. Carbohydrates can be structural and provide energy. Lipids can be structural and provide energy. Do you remember all this? Okay, and so I want you to be able to look at this diagram and to see how this is actually working. So you know that proteins are made up of monomers, and those monomers are called amino acids. Okay, and so amino acids basically have 2 carbons and an amino group. Okay, well, we can remove that amino group, and now we have 2 carbons. Okay, and some of those are just perfect to take this 2-carbon molecule, convert it into an acetyl group, and attach it to coenzyme A. And so now you've bypassed glycolysis, but you've generated coenzyme A that can go through the citric acid cycle. Meaning for every 1 amino acid you metabolize, you do the Krebs cycle 1 time. Right? For every 1 amino acid. If you've got a protein that you're metabolizing, let's say you ate just a thick steak, right? And you've got literally millions of protein molecules, and every single one of those protein molecules has like several tens of thousands of amino acids. That's a lot of times to go through the Krebs cycle, right? Which is why it's so wonderful to eat steaks, right? It's a vegetarian. Offended someone. And then so carbohydrates, we've already talked about this, right? Carbohydrates, you go through glycolysis, you go through pyruvate oxidation, you go through the Krebs cycle. Fats are pretty interesting. These fatty acid tails, we talked about this before, you've got a glycerol backbone, which is a 3-carbon molecule, and then 3 fatty acid tails. Well, you can cut those fatty acid tails off and then cut them 2 carbons at a time and shuttle them into the Krebs cycle. And then you can even take glycerol, because we don't want to waste glycerol. You can take glycerol, convert it into G3P, and then do the second half of glycolysis. And you're like, man, that's a lot. That's just a lot. That's a lot. And this is why, of all of our biomolecules types, fat provides the most energy per unit gram. Fat is about, I think it's 19 calories per gram. Okay? It's just huge. Fat is very energy dense because every single part of the fat molecule can be used to generate ATP. Every bit of it. It's wonderful. And it's delicious. Right? Which is why, I mean, that's really why we put butter on our toast, on our baked potatoes, even on our salad, right? And it's all because we're wanting to make sure we're making the most use out of the energy that's available to us. I'm right there with you. Right there with you. You also got to save up some for a rainy day, you know, because you never know when you're going to have to skip a meal for a day or 40, you know. All right, so we're going to take ourselves a lecture break, and what I want you to do is this. So at this point in the process, we've generated 4 ATP, 10 molecules of NADH, 2 molecules of FADH2. Okay? I want you to just, without using any notes, you can talk to the people around you and work together. I want you to come up with a way in which the electrons from NADH and the electrons from FADH2 can be used to make ATP. What's going on there? What is the relationship between electrons and ATP? How do you take electrons and use them to make ATP? Okay? I want you to do this. Don't use any notes. Don't look ahead in the slides. Okay? Just work with those around you. I want you to take a couple of minutes, and I want you to basically give me the relationship between electrons and ATP production. All right? 2 minutes, starting now.
And this order you have to break the area as they move, they are passing from one side and being attracted. Oxygen somehow when you see the electron so they are attracted to that oxygen, and as they pass they remember when NADH releases two electrons, it means it fills up the protons. So as those protons are going out, that passing with the protons, the electrons are.
I only have 19 emails. Some of you didn't email me.
I'm trying to make it into this one.
Oh, so it's coming.
No, mine's is separate.
So there was spark energy for every.
Oh, you still have some more time. You've got about another 30 seconds or so.
And then as they pass, they are attracting the protons, and so the protons come out of the whole matrix and the oxygen and the oxygen and I know so those are the ones attaching to the oxygen and
All right. Somebody explain this to me, because to me it seems a little strange that 22 electrons, actually it's 24 electrons, because every NADH has 2, we've got 10 of them, that's 20. Every FADH2 has 2, we've got 2 more of those. So how 24 electrons correlates to 28 ATP? Now we could figure out the amount of ATP per electron, right? But what's the connection? How do electrons translate into ATP being produced?
You need chemical energy to connect that phosphate.
You do, yeah. You've got to, if you're going to take a phosphate group and you're going to stick it to ADP, you need some chemical energy to do that. Absolutely.
That's as far as we got.
That's as far as you got. So that those electrons are in some way going to provide that chemical energy. Now we know that the movement of electrons, that's electricity. That's electrical energy, right? So we basically have a couple of options. We've got, as the electrons move, we could harness that kinetic energy to do something, or it could be just electrical energy. And so ultimately, either one of those, we're going to require to see electrons moving, right? The electrons need to move somewhere because that movement is going to fuel these processes. All right, so here's what happens. So NADH and FADH2 have electrons at an incredibly high energy level. And you know this from chemistry, that electrons at a high energy level, they're far away from the nucleus of the atom, right? But electrons don't like that. The electrons are negatively charged, the nucleus of the atom is positively charged. Electrons want to be as close to that nucleus as possible, right? Makes sense? And so electrons are going to move from places where they're at a high energy level to places where they're at a lower energy level because they can get closer to the nucleus. And if it requires them to move from one atom to another, so be it. It's a worthy move because it allows them to get closer to where they want to be, that positively charged nucleus. So, electrons will bounce from these electron carriers at a really high energy level through several complexes, dropping in energy level every time they transfer. And ultimately, they'll get to the final electron acceptor, which is our chief electron hog. And what is it? From oxygen. Okay? And they'll get to where they really, really want to be. I mean, the oxygen wants them there. Everybody wants to be wanted. So they're like, hey, if the oxygen wants me there, I'll go there because it's where I'm wanted. So this process is spontaneous. Once you generate the NADH and the FADH2, step 3 is always going to happen. Okay, step 3 is always going to happen. Now you can do step 1 and maybe not do step 3 because you've got to get those NADH into the mitochondrion, right? And that requires energy. Okay, but once you do step 2, step 3 it happens. It's spontaneous. The electrons are going to move in the direction they want to go, to lower energy levels. Every time the electron moves from one entity to another, it drops in energy level. And so this energy is used to do something. Every time the electrons drop, energy is released, and that energy is used to actively transport hydrogen ions. So every time up to this point, when we've talked about active transport, we've associated it with hydrolyzing ATP, right? You had primary active transport, that is the same pump that's pumping the material against its concentration gradient is also doing what? In primary. It's also hydrolyzing ATP. The same protein's doing both, pumping and hydrolyzing ATP. In secondary, something else was hydrolyzing ATP, and then a pump is pumping it against its concentration gradient. Well, look at this. Here we're going to actively transport this without hydrolyzing ATP. Instead, we're going to actively transport hydrogen ions by harnessing the energy release as the electrons drop in energy level. Okay? And a lot of that just goes back to kinetic energy. It's a drop in kinetic energy, and that kinetic energy, if it's going to transfer from the electrons, it's going to transfer to the surrounding materials, and that energy can be used by these protein pumps to pump hydrogen ions against their concentration gradient. And so what happens when we pump something against its concentration gradient? What do we set up? We set up a lot of potential energy. It's akin to taking a boulder and putting it on top of a cliff. What are you doing? You're setting up a lot of potential energy, right? Right? Or taking a kid on a swing and pulling them back as far as you can, right? Setting up a lot of potential energy. And then you call another kid to run by and you release the kid at just the right time. Not to hit him, because that's not good, but to just miss him, right? And have like something spectacular happen. No? And so if we start pumping hydrogen ions against their concentration gradient, we're going to set up an enormous amount of potential energy. Because now where are the hydrogen ions going to want to move? They're going to want to move back to where they were, where you just pumped them out of. And that's something just wonderful. Now, the electrons from NADH enter this electron transport system at complex 1. Okay? So NADH enters this system at complex 1, then passes from complex 1 to complex 3, and then from complex 3 to complex 4, and then from complex 4 to oxygen. Okay? The electrons from FADH2 enter in at complex 2. They don't enter in at the same place. Electrons from NADH come to complex 1. Electrons from FADH come to complex 2. Okay, and then they go from 2 to 3, from 3 to 4, from 4 to where? From oxygen. Good. Now, in between these complexes, we've got some molecules that it's just helpful to know these names. Cytochrome C and ubiquinone, also known as coenzyme Q. and these help to shuttle electrons from one complex to another. So what we find is that complex 1, complex 3, and complex 4 are proton pumps. They pump hydrogen ions against their concentration gradient. Make sense? Complex 1, complex 3, and complex 4, complex 4 and ubiquinone are all proton pumps. They are pumping hydrogen ions against their concentration gradient. Notice what complex is not part of this list. Complex 2. Which means the electrons that come from NADH go through 1, 2, 3, 4 proton pumps. 4 pumps that are going to pump hydrogen ions. The electrons from FADH2 are only going to go through 1, 2, 3 proton pumps. Do you see fewer proton pumps than this one? Those that come from NADH get one additional hydrogen ion pump to pass through. And if what that means is that the electrons from NADH are actually going to pump more hydrogen ions than the electrons from FADH2 will. Does that make sense? And if more hydrogen ions should ultimately mean what? More energy produced. Okay, so every electron from an NADH molecule is worth 1 1/4 ATP. 1 1/4 ATP. Yeah, every electron from NADH is worth 1 1/4 ATP. 1 1/4 ATP. How many electrons do we have? 20. 10 NADH that we made during steps 1 and 2, yes? Each one of them with 2 electrons, so we've got 20 electrons. Each one of those 20 electrons worth 1 1/4 ATP. What's a quarter of 20? 5. Okay, so we've got 25 ATP worth of electrons from NADH. Every electron from FADH2 is worth 3/4 of an ATP. Every electron from FADH2 is worth 3/4 of an ATP molecule. How many electrons do we have from FADH2? 4. 2 FADH2 molecules, right? One made every time we do the Krebs cycle. We do the Krebs cycle twice per glucose molecule. Yes? So that's 2 FADH2, 4 total electrons. Every one of them is worth 3/4 of an ATP. What's 3/4 of 4? 3. So we've got 25 electrons, or 25 ATP worth of electrons from NADH, 3 ATP worth of electrons from FADH2. 25 and 3 is what? 28. There's the remaining 28 ATP. Okay? And the difference there is, one, there's way more NADH produced than FADH2. Way more, right? 10 versus 2. Do you agree that 10 is more than 2?
Yes.
Do you agree that 10 is a lot more than 2? It depends on what it is, right? $10 is really not all that much more than $2. I guess $10 you can get yourself a decent meal. $2 you can't, right? Yeah.
I thought, so you said NADH creates 20 electrons for 25 ATP.
Yeah, there are 20 total electrons from NADH because we make 10 molecules of NADH during steps 1 and 2, and each one of them has 2 electrons.
Oh, okay.
Yeah. And so the big difference there between NADH and FADH2 is in the number of molecules. We make way more NADH than we do FADH2, and that every electron from NADH passes through 1 additional proton pump. Okay? Which equates to they're worth more. They're worth 1 1/4 ATP each electron versus 3/4 of an electron, or an ATP each. Yeah.
Sorry, can you remind me, how many does the FADH2 produce? How many electrons?
So it's 4 total electrons because we make 2 molecules of FADH2, each one of them with 2. So 4 total electrons worth 3/4 of an ATP each.
Okay.
Okay? And so here's just, this is in no way what it actually looks like. Now, step 3 of the process takes place in the inner mitochondrial membrane, but this is just a simplified version of it. So here's complex 1, complex 2, 3, 4, and oxygen. All right. So how is energy produced during oxidative phosphorylation? Well, we talked about that we make a steep hydrogen ion concentration, right? We are pumping hydrogen ions against their concentration gradient. Yes? Okay. And so then hydrogen ions want to go back where they came from, which is they want to go back into the matrix. And as they do, they generate ATP. So this hydrogen ion gradient is both a concentration gradient, that hydrogen wants to be where there's less hydrogen, but it's also a voltage gradient. Hydrogen ions are positively charged, yes? So if you pump a whole bunch of positive charge on the other side of the inner mitochondrial membrane, now you have a bunch of positive charge between the two membranes and very little in the matrix, okay? And so now you have two types of gradients. And so this is going to be a very, very powerful force. This force is called chemiosmosis. The movement of hydrogen ions down this gradient is called chemiosmosis. And there's only one place where they can go through. Charged ions cannot move through a lipid bilayer, right? We talked about this. Polar molecules can, but not very quickly and efficiently. Charged ions cannot move through a lipid bilayer. The only place where hydrogen ions can move through is through a complex called ATP synthase. And they're moving through fast because they desperately want to get back into the matrix because there's very little hydrogen ions inside the matrix and very little positive charge. These hydrogen ions move fast. Enormous amount of kinetic energy that can be used to take inorganic phosphate and attach it to ADP. This energy of chemiosmosis, in addition to making ATP, as those hydrogen ions move through ATP synthase, that energy can also be used to actively transport materials into the mitochondria, to actively transport pyruvate into the mitochondrion, to actively transport other materials into other proteins that function in the mitochondria. Okay, does it make sense? If you start building up a steep gradient, again, think of it like taking a boulder and pushing it to the top of a cliff. Okay, now you've set up a lot of potential energy. When that boulder's released, man, is it going to move. And that's what we've done by pumping these hydrogen ions against their concentration gradient. We've set a boulder on a cliff. And when it's allowed to go, it's going to go, and it's going to go fast. And these hydrogen ions are going to go through ATP synthase, and as they do, they're going to fuel the production of ATP. Did I answer your question?
Yeah, I was going to ask where they go.
Yeah, they can only go through ATP synthase.
That's how they release the gradient?
Yes.
Okay.
Yep. You bet. And so this we've already mentioned. Each NADH molecule is worth 2.5 ATP because it has 2 electrons that are each worth 1.25. Right? 1.25 times 2 is 2.5. And then every FADH molecule generates 1.5 ATP because it's got every FADH2 has 2 electrons that are each worth 3/4 of an ATP. And why is there a difference? Why does NADH generate more ATP, or why does an electron from NADH generate more ATP than an electron from FADH2? Yeah, Davies.
Because it goes through an extra
Goes through an extra proton pump. It pumps some additional hydrogen ions. Good. Love it. So the ATP generated during cellular respiration is about 224 kilocalories per mole of glucose. So the amount of ATP generated during cellular respiration, all 3 steps, is about 224 kilocalories per mole of glucose. If you burn glucose, it releases 686 kilocalories per mole. Okay? So we're like, all right, that's 3 times, more than 3 times the amount of energy. This is why we don't burn glucose inside of our cells. Okay? Just too much energy being produced. But on top of that, if we're only getting this amount of ATP, it tells you that there's additional heat being released in this process, right? Which is good for organisms like us. Why is producing heat in this process good for organisms like us? We're warm-blooded, we're endothermic, and the bulk of our heat comes from our metabolic rate. Okay? That's cool, right? Now baby humans, their heads are so big compared to the size of their body, right? And the head is where we lose most of our heat. And so babies lose way more heat than we do, which is why if you've held a baby, they always feel warm. And then you check their body temperature and it's basically the same as yours. And you're like, why do they feel so warm? Have you had that phenomenon? You held a baby before? They always feel warm. And it's because they lose a lot of heat. But it's also why their metabolic rate is way higher than ours. Otherwise, they couldn't maintain body temperature. And that's why, by the time like a kid's 6 to 8 months old, it's consuming like 1,200 calories a day. Which if you're like, okay, we've got this 20-pound human consuming 1,200 calories a day. If we were to extrapolate that out to a 170-pound human, you're like, man, that's a lot of calories, right? I mean, Michael Phelps, when he was training for the Olympics, consumed 17,000 calories a day. But that's not normal, right? That's because he was swimming like 30 miles a day. Probably not that much. I don't know how long it could take you to swim 30 miles. That's a big number. But he was swimming a lot. Oh, and The Rock, when he trains for a movie and has to bulk up, I mean, he consumes like 15,000 calories a day. Okay? But he's also got to put on like 30 pounds of muscle mass in a couple of months for some role, All right. So if you were to look at this, what is this process? Explain to me what's going on here.
Glucose is turning into ATP.
Uh-huh. So what is it generating here? What are we showing here?
ATP.
This is the overall process of the first 2 steps of aerobic respiration. Right? We take glucose and we take 4 ADP and 10 NAD plus, 2 FADs, and we generate 4 ATP. That should clue you into that this is the first 2 steps, right? First 2 steps after they're done, we've generated 4 ATP, 10 NADH, 10 hydrogen ions, and 2 molecules of FADH2, and 6 molecules of carbon dioxide. Okay, so this reaction shows you the first 2 steps of aerobic respiration. This is a nice diagram showing you a more realistic picture of what the electron transport chain or oxidative phosphorylation looks like. Here's a proton pump, proton pump, proton pump. Okay. The electrons from NADH come here, which allows us to pump some additional hydrogen ions. Here's ATP synthase, the only place where hydrogen ions can actually move through, and they will really fast. And that kinetic energy can be harnessed to basically turn this mechanism and take ADP and an inorganic phosphate and make them into ATP. Okay. Now this whole thing will only work if you've got the final electron acceptor, which is oxygen. And so the electrons, here are the electrons, boom, 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 and then here finally going to oxygen to produce water. Okay? And so this whole process only works if oxygen is present. Yeah. Okay. So here some people did some experiments to see if we could use a different type of energy to actively pump hydrogen ions against their concentration gradient. So they used light energy to do that, to actively pump hydrogen ions against their concentration gradient. And then basically the same thing happens. That hydrogen ions move down their concentration gradient through ATP synthase to make ATP. Okay? And so here's a little bit of a picture of what you might anticipate in the next chapter when we talk about photosynthesis. I mean, there's this picture here with plants, but of actually using light energy then to fuel this process, to fuel pumping hydrogen ions against their concentration gradient. All right, last question we need to deal with, and we'll do this one relatively quickly. What if oxygen is not present? What if oxygen is not present? First of all, can we do steps 1, 2, and 3 without oxygen? No. I told you the whole thing only works if oxygen's present. Okay? So we can only do glycolysis. Okay, so if oxygen isn't present, all we can do is glycolysis. In glycolysis, we produce how many ATP? 2. And we also produce 2 molecules of NADH. But now if we can't do step 3, there's nothing for that NADH to do with those electrons, right? And so now we're going to start building up a whole bunch of NADH with nothing to do with it. That's not good, right? When you start building up product and have nothing to do with it, bad things happen. Okay, so we really have 2 problems that arise when oxygen isn't present. 1, we're not producing nearly as much ATP. 2, we're building up a whole lot of NADH. Okay, so you should anticipate that these electrons from NADH are probably going to go somewhere so that we can restore NAD plus and continue to go through glycolysis. Yep, glycolysis continues to generate ATP, not much, but some. And then pyruvate would be another thing that would start to accumulate if all we were doing is glycolysis. So pyruvate needs to get converted into something, and we need to have NADH give its electrons to something. The beauty here is NADH gives its electrons to pyruvate, converting it into either lactate or ethyl alcohol, depending on the type of fermentation present in that organism. For the yeast that we worked with in lab, when they ferment pyruvate, they generate ethyl alcohol, which is why we use yeast to make beer, to make wine, to make any other alcoholic beverage, because when yeast ferment, they produce ethyl alcohol from that pyruvate. So you feed a yeast sugar and you get ethyl alcohol. When we ferment, we do not produce ethyl alcohol. If we did, it would make running marathons really complicated, right? As you run a marathon and you start to lose oxygen from your muscle tissues and you start to go through fermentation instead, we produce lactic acid or lactate, which drops the pH and gives you the cramps unless you've increased blood flow to your muscles and you can flush that out. But if we fermented ethyl alcohol, as you went on like running a marathon, you would become incredibly intoxicated and you would very quickly die of alcohol poisoning. Okay? Might make watching a marathon more interesting, but it would make running a marathon very, very complicated. Okay? But either way, whether you ferment lactate or you ferment ethyl alcohol, the NADH made during glycolysis gives its electrons to pyruvate. So here's lactic acid fermentation. And so here we're making NADH during glycolysis. That NADH gives its electrons directly to pyruvate, generating lactate. And then that lactate will diffuse out of the cell. All right? And then here's alcohol fermentation. Again, the NADH made during glycolysis, it has to give its electrons to something. It gives its electrons not directly to pyruvate, because before pyruvate is converted into acetaldehyde, and then it gives its electrons to acetaldehyde to generate ethyl alcohol. All right? So again, 2 problems if all you're doing is glycolysis. Way less energy produced, and you're building up NADH and pyruvate. Fermentation eliminates not the very little energy produced, but eliminates the buildup of NADH and the buildup of pyruvate. Yeah, Levi.
So why in our muscles does this happen when we start to really vigorously exercise?
Yeah, because you're just metabolizing more quickly than you can get oxygen to those muscles. But as you get in better shape, your blood flow to those muscles increases. So you do a better job of getting oxygen to those muscles, and you do a better job of washing the lactate or lactic acid away. Yeah. All right. Yeah.
What's the final destination for lactic acid?
Lactic acid, ultimately, you excrete it in your urine. Yeah. And so the ultimate fate of it is it depends on where the ultimate fate of your urine is. Yeah. Yes. Yep. Absolutely. Yeah. The better shape your body's in, the better you'll be able to get oxygen to your muscles. Yep. Absolutely. All right. We're out of time, and we're out of material.